All right, everyone, second part of our video this week. We want to think about, again, like this diversity, right? Diversity of life on the planet. And we're talking about mostly domain bacteria at this point. So here we have arrangements of flagella. Again, a very important characteristic to know what kind of bacterium you're intentionally identifying. So you see, again, in the figures A, B, C, D from left to right. Mono means one, so monotrichous. Trichous comes from trichum, which means hair. So one hair, so one particular flagella. We can have peri. Peri means like around the perimeter, around the edges. So peritrichous means that you have flagella all around the outside, few or many. Lophotrichous, essentially we have flagella on just one end or one pole, if you will. And then we have amphitrichous, so one on either end. And look at that. Isn't that a beautiful, beautiful spiral shaped cell? Okay. So in your study guide this week, we talk a little bit about how bacteria move in this essentially run and tumble. So why would a bacterium need to move? Well, we talk about chemotaxis, so either we're moving toward or away from a chemical signal. Positive chemotaxis means you move toward a favorable signal. So that could be like oxygen or sunlight or nitrogen or something like that. I'm thinking about waters or puddles, or even if they're like living in your body, they want to move where they live most ideally, right? Negative chemotaxis means you're moving away from something. So maybe like if my immune system is launching a full-on defense, I know we have one of our second lines of defense, we use white blood cells. And so we have a white blood cell called a phagocyte, and it gobbles up bacteria. Now, if I were a bacterium and I could essentially respond to chemicals made from the immune system, then I would move away from that immune system. So that would be negative chemotaxis. So when there are no chemical, okay, this is weird. When no chemical gradients, okay, just take off that first phrase. Essentially, when organisms are moving, without needing to have some direction. So when it says no chemical gradient, so when it's just living its life, it is going to do this run and tumble. So you see in figure A, this little pink bacterium, it's rotating its flagellum counterclockwise, and it's just like moving along. But then essentially it stops, it reverses the flagellum. So now it is going clockwise, and it causes the cell to stop, and it tumbles and it can change course. So if I'm peritrichous, essentially I'm going to coordinate the movement of my flagella so that I can move in my direction. And when I stop, all the flagella then are going to like essentially work on their own, and it causes the cell to tumble. Okay, so something to think about. All right, here's our fimbria. I know we talked about it in the first part of this video. Small bristle-like fibers. Helps these organisms stick to a host or stick to potentially the surface that they're trying to grow on. So that's important. And then it also allows them to hang out with others more easily. So safety in numbers. All right. In the previous video, I remember telling you that bacterial sex is called conjugation. It kind of sounds like conjugal visit, but conjugation. And you see here, we have these pili, so these long strands between the cells. And they're trying to show us that the fimbria are these little tiny bristle type things. You really have to stare at them. But I find the most interesting thing on this slide is the pili, because they are connecting from one bacterial cell to another, often of the same species. That's usually what we think about in these things, but some species have the ability to reproduce with ones that are really closely similar. But essentially what happens in these pili is that a copy of that circular double-stranded piece of chromosome is going to be exchanged between the two organisms. So we call it conjugation because you each contribute something to the other one. And they do that through those special extensions called pili. Okay, so To me, the most difficult part of this chapter is like sorting through the S layer and the glycocalyx, and then learning about different kinds of lipopolysaccharides. So we will try our best to like slow this down to understand it. All right, so an S layer, single layers of thousands of copies of a single protein linked together like tiny chain link fences. And this S layer is going to protect the bacteria in hostile environments. So I would not have an S layer if my life is pretty good and I, don't really need to go into that mode. So the S layer may or may not be present. I got a cat meowing. The glycocalyx. Glyco means sugar, and calyx means coating. So it is a sugar coating. It can have some proteins associated with it. So just be thinking sugar coating, but also maybe some proteins. So when we have polysaccharides, this is repeating sugars. Okay, so the glycocalyx can essentially be organized in one of two ways. So we have a slime layer, which is a loose layer, and it's just, kind of just helping that bacterium deal with changes in the environment. So it protects against water loss or nutrient loss. But a capsule, man, this is like really seriously getting more defensive. So it's more tightly bound, denser, thicker, it's sticky, and it's really going to give this bacteria some properties, that it can succeed with, if you so to speak. Now, when I'm looking at petri dishes of bacteria, that capsule is going to essentially give my colony a glossy or slimy appearance. So you can actually see them with the naked eye on cultures of bacteria. Now take a look at this. So here we have some encapsulated bacteria. So that capsule is there, and take a look. It is not, in figure B, it is not absorbing the stain. So you actually see kind of a halo around those bacterial cells. And again, on the agar plate, as we see in the figure to the lower left-hand side, it's forming kind of a slimy or mucousy looking colony. Okay, so the glycocalyx, again, can be just in this thick, rigid outer coating, and is formed by many pathogenic bacteria, because this is going to give it a hold in its host. Okay, and oftentimes it's going to protect us protect them even from our white blood cells trying to gobble them up and get rid of them. So we know that these capsules are pretty important to those bacteria that are pathogenic. And we know that not all bacteria are pathogenic. So we have, good bacteria. In fact, we have so many symbiotic bacteria in our bodies that are protecting us against the bad bacteria. Out in nature, how's decomposition occurring? It's occurring because bacteria are colonizing and breaking down that organic matter in those in that organic matter. Excuse me. So bacteria, there are many beneficial bacteria not causing health issues. So biofilms, as I mentioned earlier, that glycocalyx really can help cells stick together and form like a super colony. Biofilms, we have biofilms on our teeth. So when we start getting plaque buildup on our teeth and needing to go to the dentist, essentially it's these individual cells that are sticking together with other cells and forming this like very hard, rigid layer it's hard to get off your teeth. And that causes, they release acids, and that's what causes our cavities or dental caries. Now, you probably know that in the healthcare field, we're always concerned about aseptic techniques, and like anytime you pull out, if it's IV tubing or if it's perhaps catheter and so on, like you have to learn the sterile technique to insert these things, because we know that they are very good surfaces for biofilms to occur. Now, the medical industry has for the past 10-15 years, has been trying to create new plastic so that it becomes impermeable to bacteria. So you might have heard of Microban, something you can spray on your sandals, especially if you wear like Tevas or those kinds of shoes where you're getting in water and your feet sweat, kind of prevents that from occurring. So here's a step-by-step picture showing us the development of biofilm, and you can see that biofilm becomes a place that other bacteria can live as well. And so now you have essentially a community of bacteria in your biofilm. All right. Next up, we have the cell envelope. It lies outside of the cytoplasm. So this you already know because we called it, because it includes the cytoplasmic membrane, or that plasma membrane. So this is really going back to that inside out. So you have your cytoplasmic membrane, and then we have the cell wall. So that's all the way to the inside. Well, let's take a look at our last topic today, the comparison of gram-positive and gram-negative envelopes. So the envelope, again, is these three parts, or these three features of the cell. And we can we can break down our domain bacteria by how they react to the gram stain. So you see on the left, we have this really thick purple layer, and this is called our peptidoglycan layer, peptidoglycan. If you look to the right in our gram-negative bacteria, we essentially see there's a thin layer, but there's also like this outer membrane layer, okay? So different structures, but what I want you to know now is positive has a thick peptidoglycan layer and negative has a thin layer. And yes, there are some other features here. Here's a little diagram. So if you have the inside of the cell down here at the bottom, and then first you're essentially going to have that plasma membrane, or also called the cytoplasmic membrane. Then we're going to have this outer layer, this wall, if you will, or this envelope, this wall. And it's called peptidoglycan layer, so it's thick here, and over here it's thin. And then we see that there's an extra layer of lipopolysaccharides out here on the outside of this gram-negative cell, and it has something we call lipid A. Now, when gram-negative bacteria invade your body, that lipid A can give you some of those symptoms that you have of sickness. So just something to be aware of. Why do we have these cold symptoms? It's not all your immune system launching. Sometimes it's toxins that are released by the organisms we are exposed to. So, excuse me, you can pause on this slide. I don't want to run out of time, but you see, obviously, the peptidoglycan layer is very thick in this organism, and we don't see lipopolysaccharides or LPSs to the outside of the peptidoglycan layer. In our gram-negative, we see it's just 1 to 3 nanometers, so it's so small. And these organisms are susceptible to what we call lysis, or bursting, because they don't have that big, thick layer. And then, of course, the outside layer has the lipopolysaccharides. You can read there, endotoxins, that lipid A can cause fever and shock in gram-negative pathogens when they invade your body. Okay, so let's take a look. So this gram stain, this is a this is going to be a staining mechanism that we do in lab, either online or on campus, but essentially this is how it works. We are going to adequately prepare our slide or our sample. So I'll have bacteria in lab. Online people, you'll have your bacteria that you use in your virtual lab. And essentially we are going to make a slide of the organism. We are going to allow it to dry, air dry, and then we're going to fix it to the slide by putting it through a flame of a Bunsen burner about 3 times. Then we can stain it. So we first stain it with crystal violet, okay? You keep it there 45 seconds or a minute, okay? It's going to absorb. Then we're going to rinse that off, and we're going to add gram iodine. Gram iodine has the ability to attach to the crystal violet. So in my gram-positive cells, that crystal violet is stuck inside. Actually, it's still in the lipopolysaccharide, it's still in the peptidoglycan layer, okay? So that crystal violet is still there. We stain it with gram iodine, and now the iodine is going to attach to the crystal violet, and now it contains or stays in the cell. Gram negatives, well, we're going to go ahead and stain it with crystal violet. We will rinse it. There'll be a little bit there, but then we're going to put gram iodine on it, and because that peptidoglycan layer is so thin, it's not absorbed. So all of that is just essentially not sticking in the cell. Now we rinse the cells with alcohol to truly, we call it destaining. And then we add red dye so that any colorless cells now appear red. So if you're following along with me, gram negatives are going to be pink because they didn't turn purple, because that didn't stick in the cell, and gram positives are going to be purple.